How's it going, everyone? It's Chow here. So a couple of you told me that you enjoy these videos that accompany my lecture slides, so I'm going to make more of these. So for today, we're going to talk about cell membranes, which is just an absolutely phenomenal topic. But let's go over some of the specificities as well as some of the basics to get an overall understanding, and hopefully by the end of this video you may get a little bit better of an idea as to what you need to study for the second exam. So let's get started. Alright, so with cell membranes and everything else, the key thing to think about is this concept of the fluid mosaic model, where nothing really stays stationary. Things are very mobile, things move around. Membranes are not static, they're very dynamic and they're active, and the little proteins and the little phospholipids will move around in the membrane, hence this concept of a fluid mosaic. What's really interesting, however, of course, is the fact that despite having this ability to move, it's generally slower in terms of movement than, say, solutes in the cytoplasm. I guess you can think about it in that sense. So it's not very stationary, and that has many implications for the cell. One thing that perhaps is a very strong implication is the fact that there are lipids that can move around, but there's restrictions to that movement. And one restriction that we talked about is that lipids don't really flip from one side of the bilayer, so over here, from this side of the bilayer to this side. So they generally only stay on their own specific side, which it makes sense in many ways. So they only stick around, again, on their side of the bilayer. Hope that makes sense. That's not a super important point, but it is something that is somewhat critical to our understanding of the cellular membranes. Now, there are lots of membrane proteins in the cellular membrane, not just little phospholipids floating around, but also different types of proteins. So there are basically two main types of proteins that we talked about. We talked about integral membrane proteins as well as peripheral membrane proteins. And the main difference is where they're situated. So as you can see here, integral proteins span the entirety of the cell. And there's a domain that is hydrophobic, and they're embedded in that hydrophobic region of the cellular bilayer, which again makes sense. Now, there are also other proteins called peripheral membrane proteins, and these peripheral membrane proteins are basically found on one end or the other end of the cellular bilayer. And the reason why is because they often like to interact with hydrophilic or polar heads of phospholipids. So if they like to interact with hydrophilic situations, think about where they're going to be located. Well, definitely not here in the center over here where everything's hydrophobic. They're more likely to be found on one side of the membrane or the other side of the membrane. Again, the heads of these phospholipids are pointing towards liquid, so aqueous solutions. Perhaps you can think about it down here as the inside of the cell and the outside of the cell there. Liquid situations, aqueous situations, and therefore there are going to be polar interactions. And so peripheral proteins like that, and they point towards they're either found at the top or the bottom of that bilayer next to those phospholipid glycerol heads. So I hope that makes sense. So moving on from there is this other concept, and that is the idea of this lipid raft. So membrane proteins have a tendency to actually bind to special lipids to form these rafts looking thingies that float kind of around at the tops of these membrane bilayers. And there are kind of two special lipids that allow this situation to occur. One of them is sphingomyelin, which kind of resemble normal phospholipids. It has a polar head with choline and has two nonpolar tails, as well as this other perhaps well-known lipid called cholesterol that kind of work together with sphingomyelin to make this lipid raft. Of course, you can always think about it with this particular formula over here that Dr. Leckler gave us, and that is lipid raft is basically sphingomyelin plus cholesterol plus some kind of a protein attached. So you don't really need to know that much about lipid rafts, but know that they exist and know what exactly they are. So lipid rafts, pretty straightforward, pretty cool. But what's perhaps more important is this idea of cell adhesion and cell recognition. Individual cells have to somehow communicate with one another, and there are many ways they can communicate with one another. One way is with all these little tiny proteins that have some kind of extra thing attached to them, if we want to be very vague about it. But cell-cell recognition uniqueness is often defined by unique lipids and unique proteins. So one thing that we often talk about is this idea of glycolipids. You might have heard this term being thrown around sometime in the past. Glycolipids, as the name suggests, are simply carbohydrates that are attached to the lipid and attached to the lipid cell surface. So, pretty interesting. Perhaps more interesting are the glycoproteins, which are carbohydrates that are attached to an amino acid, for instance, on the surface of proteins. And then there are also proteoglycans, which are simply extensive carbohydrates that are attached to proteins, so very, very big carbohydrates. So very, very important things that are needed for cell-cell recognition. Now, that's cool and all, but what about adhesion? What about that connection between cells? Well, there are different types. There are actually 3 different types of cellular adhesions that we talked about. The first one that we mentioned was this idea of a tight junction. So tight junctions are simply membranes that are tightly packed together and kind of interwoven. Think of it like stitches, as mentioned here on the slide. And then there are also proteins. These proteins form these tight junctions, which allow for this concept of intercellular space between these 2 cells. So they are very tightly connected in terms of this particular type of cellular adhesion. Tight junctions, they're pretty important, but I think the next ones are probably a little bit more important in many ways, because we talked about this other thing called desmosomes. Now, desmosomes are like loose seals. What's very interesting about desmosomes is that they link adjacent cells tightly, but they allow materials to kind of move around them in the intercellular space. So there's this protein mesh that produces these desmosomes, but they still allow some sort of movement within that intercellular space. Whereas if you think about more tight junctions, it kind of bars the movement of many of that materials through the space between the cells. So that's something to keep in mind. And last but not least, we have this thing called gap junctions, which are just like protein tubes or channels between the 2 cells, and they allow substances to travel through. So they let these adjacent cells communicate very quickly via these protein tubes. So those are the 3 types of adhesive properties that connect different cells together that we mentioned. So perhaps something to just keep in mind. So this next concept that we talked about was the extracellular matrix found in animal cells. And this allows cells to kind of have this interaction between the inside and the outside. So the extracellular matrix is just this protein mesh that's found on the outside of the cell. And what's really interesting perhaps about the extracellular matrix is that it's actually connected to the inside of the cell via these integrin proteins over here. And then in the inside of the cell we have the cytoskeleton, which is a protein mesh of actin fibers. And this is very incredible because there's this sort of stability, and this stability is fairly important to cellular function and cellular structure. And of course, something that we also talked about was that there are actually 24 different types of integrin proteins that kind of connect the extracellular matrix with the inner cytoskeleton. So lots of different proteins, but thankfully you don't have to know any specific one. Just know that there are integrin proteins that link the outside of the cell to the inside of the cell. So let's move on to something else. And this is something that perhaps we kind of beat the dead horse again and again and again and again, but it's something that perhaps can be a little confusing to an extent, and that is this idea of diffusion. Now, diffusion is nothing more than the process of just random movement of certain molecules towards a certain equilibrium. So we have this formula over here of delta G equals delta H minus T delta S. And then we talked about how diffusion is driven by entropy. Lots of scary things within this molecule. But the thing that's probably most important in this slide is this thing over here. If there is a concentration gradient, molecules will move down the concentration gradient. So when there's diffusion happening, you're trying to equalize concentrations. So if there's a high concentrations of solutes in one area, and there's nothing blocking those solutes, then those solutes will most likely try to diffuse across the aqueous solution to balance out and equilibrate and find that equilibrium. But of course, it's very important too, because even at the equilibrium, particles and molecules are still moving. But there's no net change in the distribution because there's just a net movement, and the amount going to this side is the same as the amount going to that side. So it all balances out. So diffusion, seemingly straightforward of a concept, but with many, many different implications. One of those implications are these two scary, scary parts. And that is the difference between the two types of transport. So we talked about this in class pretty aggressively and went into a lot of detail in regards to it, so I'm going to go into detail in regards to it as well. So we have passive transport as well as active transport. And passive transport really is just diffusion that doesn't require specific cellular input of energy. Whereas with active transport, that requires energy, because in many cases of active transport, you're trying to push something against the gradient. So if you have a situation where you have a lot of solutes on one side of the membrane and then very little solutes on the other side of the membrane, you're actually trying to push solutes from the side that doesn't have a lot of solutes into the side that has a lot of solutes. So in order to do that, that's not very energetically favorable. So you need energy to kind of push it to the other side. So there's that. So let's talk a little bit about the individual types of diffusion, since we talked about them in class. There's this thing called simple diffusion, which, as the name suggests, is probably the most straightforward one. So we have simple diffusion, which really is kind of the traditional sense of diffusion that we think about when we say the word diffusion. But there are some key points about simple diffusion that might be worth noting. For one, it's often small or uncharged molecules that diffuse through a phospholipid membrane. So things like water, oxygen, carbon dioxide, alcohol, yes, alcohol like ethanol, good stuff, right? And then of course glycerol can easily diffuse across the membrane. Now there are also some other hydrophobic molecules that can diffuse through the membrane, like certain hormones like estrogen. But if you look at this picture here, this is probably a good representation. No proteins present in this section of the membrane, yet you have certain molecules that are able to sort of flow from the outside of the cell here, as represented, into the cytoplasm. As time moves on, more and more of the solutes diffuse through until there is an equilibrium that's reached. And again, when the equilibrium is reached, there is still equal exchange of materials. It's just happening at a net rate. So it doesn't ever stop in terms of diffusion, it's just happening at a net rate, so you don't see this extreme change. So there's that, simple diffusion. Pretty straightforward. Next, we amp it up a little bit and make it a bit more complicated with facilitated diffusion, which really requires sort of these things called carrier proteins, which are just simply proteins that are used to transport molecules. So One thing that is often commonly used in biology as an example is the glucose transporter, which basically brings glucose from an area of high concentration, often outside of the cell, into the cell via this interesting movement as shown over here. And so the protein facilitates the movement of glucose from one location to the other. You're still going with the concentration gradient. Normally glucose cannot just go through the membrane, but with the carrier protein, it can. So again, think about it. Facilitated diffusion of glucose, it's still going with the concentration gradient. It's not going against it. It's just simply needing help in the process because glucose is too big of a molecule. So keep that in mind as we move on. The next thing that we talked a little bit about was this idea of a gated channel. So gated channels are also in a way facilitated diffusion, but generally speaking they only open as a result of some kind of stimulus. The stimulus can often be chemical, it can be electrical, it can also be sort of mediated by a ligand, so a stimulus molecule as shown here. And normally when this stimulus molecule, this ligand, is not binded, the channel protein here is closed. But when this ligand is bound to this protein, the channel protein opens and thus allows the little solutes to go through. So one example of this is the voltage-gated sodium channel. And so in that case it's the voltage gating, where the sodium ions are transported, but only when the membrane voltage or the potential reaches a certain level. So that's a situation where there's a certain gate that kind of blocks the diffusion from happening, but it can be activated via some kind of regulatory sequence and a regulatory molecule or a shift in chemical or electrical potential. So that's gated channels, another type of facilitated diffusion. And finally, we have active transport, which is probably the one that might get a little bit more complex because of the fact that it requires energy. Because active transport is going against the concentration gradient, which is not energetically favorable. So it requires the energy input. Oftentimes, that's ATP hydrolysis. And it uses that ATP hydrolysis and that energy from that ATP hydrolysis to couple the transport of certain molecules. There are many types of these active transporters. There's a uniporter, which basically transports one substance one direction over here. So this blue solute is going in one direction. In a symporter, it's actually two different substances going in the same direction. And finally, we have an antiporter, which basically transports two different substances in opposite directions. So this brownish-looking molecule is coming in, while this reddish-looking one is heading out. So that's active transport, and that pretty much sums up most of the diffusion of solutes. And I guess I will include osmosis within this particular section as well. So osmosis is just the movement of water. It's also diffusion, but you're moving water molecules instead of solute molecules. So with osmosis, there are kind of three big terms that we talk about. The first one's isotonic, which just means that the concentration of solutes on the inside and the outside are equal. There's hypertonic, which means that the solution is going to have higher amounts of solute content than the solute content inside a cell, for instance. And finally, we have hypotonic, which is just kind of the opposite of hypertonic, where there's an instance where there's more water on the outside of the cell than on the inside, or in other words, there's a higher solute concentration on the inside of the cell than the outside. So the picture that I showed over here, I think, is a good way to describe these three different terms. So hypertonic, you have more solutes outside here than inside the cell. So naturally, if you limit the movement of solutes, for instance, sodium, let's say there's it's sitting in a salt bath, so a bunch of salt water outside over here in the blue, and you have a bunch of red blood cells. What happens is the water molecules can move between the inside of the cell through the membrane to the outside and vice versa, but the solutes cannot. So in a hypertonic situation, water will move out of the cell, and therefore the cell will shrink. In opposite, there's a hypotonic situation where you can imagine it's a red blood cell sitting in a vial of very fresh water without any solutes. And so the solute concentration inside the cell is going to be much higher than the solute concentration on the outside of the cell. So what happens? Well, naturally, water is going to diffuse from the outside of the cell to the inside of the cell, because again, hypotonic is a situation where the inside of the cell has a higher concentration of solutes than the outside. So again, water is going to move into the cell, and if that happens too frequently to try to equalize the equilibrium, you might get an instance where the cell actually just explodes and bursts, which of course is not good. Finally, an isotonic situation, which means that the solute concentration on the inside of the cell and the outside of the cell is exactly the same. And when that happens, you have equal exchange of water going in as well as water going out. Keep in mind that in an isotonic situation, there is still movement of water molecules in a cell that has a membrane that's impermeable to certain solutes. So that exchange is still happening. It's just that it's at an equilibrium, so therefore you don't see any certain changes in the amount of water that's going in versus the amount of water going out. It's just an equal exchange despite the fact that it has reached equilibrium. So that's just something to keep in mind. Water and diffusion still occurs in an isotonic situation. So I guess finally we can just summarize everything out into this particular table that's also found in your textbook, looking at different types of diffusion. So we have simple diffusion, diffusion through channel, facilitated diffusion, as well as active transport. I should have probably included this before the osmosis section, but I think it was better to talk about osmosis beforehand and then just summarize everything here. But this is a good thing to look through and understand whether energy is used, what's the driving force, whether membrane proteins are required. And I guess probably the most key thing here on this page is likely active transport, where you have yes to all these questions, versus simple diffusion, which is more or less no to most of these questions, I guess, except for driving force there. So I hope you found this video helpful. This is supposed to go alongside with some of the things I lectured over, and I will see you in perhaps the next video. Take care, everyone.